Okay, so section 1.3.1, chemical transformation, aka chemical reactions. So these two terms are interchangeable, chemical transformation and chemical reactions. First, we're going to go over what is the difference between a chemical and physical reactions, because both are going to be important for the remainder of this class. A physical reaction is simply a process where the identity of the participants in the process don't change. It's just their position is going to change. So, for example, a binding reaction, A plus B. These are two molecules that bind together and give you AB. Now, on both sides, you still have the two molecules AB, but their position to each other has changed. Instead of A being alone and B being alone, now they're linked together through weak interactions. But this is reversible, meaning that it can simply be reversed too. So this is a physical reaction. Now, in the case of a chemical reaction or chemical transformation, for example, A plus C, it gives a whole new molecule, for example, D. So the chemical. So in this case, what we see on the reactant side is different than what we see on the product side. A and C are two molecules, and D, it's its own molecule. Of course, the atoms have changed the way they're covalently bound to each other, but you have a whole new molecule on the product side than what you had on the reactant side. And that is the difference between a physical and a chemical reaction. Now, condensation and hydrolysis reactions. So most reactions in a cell are going to be of either one of these two types. It's either going to be a condensation reaction or a dehydration reaction. Either name works, they're interchangeable terms, condensation or dehydration, and hydrolysis reaction. Now, condensation reaction is going to link two molecules together. So you can see that we have molecule A here and molecule B, and they are covalently linked together in the product side. And an H2O is kicked out. The H2O is also a product of this reaction. It's just shown this way sometimes when we draw stuff in biology. In the case of a hydrolysis reaction, you have A and B, and an H2O comes in, so the H2O would be a reactant, and you break that covalent bond between A and B. So an example of this, if we're just not going to have some generic stand-ins for molecules, we're going to use an example of two glycines being covalently linked to each other through a peptide bond to form a dipeptide. And these two glycines will give us the dipeptide. Now, where does the H2O come in? Because this is going to give us also an H2O. The H2O comes from this oxygen and two hydrogens from the NH3+. Now, in the case of a hydrolysis reaction where that bond is broken, you come from here and you go in this direction. Two separate reactions, hydrolysis and condensation. Now, it's mentioned here that condensation reactions are energetically unfavorable, and that hydrolysis reactions are energetically favorable. So one way to think about it is building things in a chemical reaction where you're taking two smaller molecules and making a bigger molecule is energetically unfavorable, and it will require a condensation reaction. Whereas taking a bigger molecule and breaking it up to smaller molecules is energetically favorable, and it requires a hydrolysis reaction. All chemical reactions supporting life, whether within or outside the cell, are catalyzed by enzymes. There is no exception to this rule. Every chemical reaction in the cell is catalyzed by an enzyme. And what is catalysis? Catalysis is nothing but increasing the rate by which a reaction will happen. And we'll cover all this stuff, favorability and catalysis, in more detail later, just introducing the terms now. So what are enzymes? Enzymes are biological catalysts which increase the rate of a chemical reaction but are not consumed by the reaction. So this is a definition right here of enzymes. So here we have a picture of an enzyme doing its work. It's a generic picture, but we can draw it differently too, but let's go over this picture first. we can see that the enzyme right here comes in and it has an active site. So here's the active site of the enzyme. It binds the substrate. The substrate is nothing but a molecule in the reaction. So in this case, the reactant is molecule A and the product is molecule B. So if I was going to write this reaction out, it'll be A goes to B. So the active site binds the molecule A, the substrate for the enzyme. So the molecule, the reactant that binds to the active site and is transformed to another molecule, because this is a chemical transformation, is called the substrate of that enzyme. And it forms the enzyme substrate complex right here. Then catalysis happens and the substrate is transformed to the product right here. And then the product is released by the enzyme right here. And the enzyme can go and pick up another substrate in this direction. Right? So this whole thing can be written out like this. Enzyme plus substrate gives me the enzyme substrate complex, which then gives me the enzyme product complex, which then gives me enzyme plus product. And then the enzyme is free now and it can go to pick up another substrate. So the enzyme was not used in the reaction. The only thing that was transformed was a substrate was transformed to the product. So the above picture is representing a very favorable reaction, and we'll go over what that means later. An enzyme will catalyze a single reaction. This is called enzyme specificity, meaning that an enzyme will only catalyze a specific reaction. It will not catalyze other reactions. Under certain conditions, an enzyme can catalyze a different reaction, but it is rare, therefore dismissed as it has no biological consequence. And this is called an off-target effect. So, under certain conditions in the laboratory, in a test tube, you can make an enzyme use a different substrate than it doesn't normally use in the cell. But that is not important to us because we're studying cell biology. So, that is irrelevant to the discussions that we'll be having. A cell controls the chemical reactions within it by controlling the activity of its enzymes. So maybe there's a couple of things that we need to talk about before we move on with this statement. So when we're talking about the chemistry that is happening within the cell, there's a few things that we can assume. One of them is that the temperature is going to be a constant. So in the case of our cells and many mammals, 37 degrees C. So when we're talking about chemical reactions in a cell, we don't really care about what will happen if there's a variance in temperature. Because if there's a variance in temperature, then things will go wrong, especially if the variance is very high. But even if the variance is very low, just a few degrees in either directions, the cell will stop functioning in the correct manner, and that can lead to all kinds of problems for the organism. Now, what else is fixed? Now, of course, pressure is fixed. They're always going to be under 1 atmosphere or so. So we're not going to even discuss that because that's not going to change. All the reactions are going to happen in a solution where the solvent is water. So there's plenty of water around. The reactions will happen in an aqueous environment, so that's fixed too. What else is different than what you would see in a chemistry lab, for example? The concentration of all molecules and ions in a cell is kept pretty constant. So all the reactants, their concentrations are at a pretty constant level. So if you're in an environment like this and you're trying to do chemistry, so this is our environment right here, and the cell needs to do some chemical reactions to maintain itself, well, you cannot control the concentration of reactants. I mean, that's one of the things that you do in a chemistry lab. If I want a chemical reaction to happen, I get my reaction pot, I add the reactants that I want, then I know will give me the product that I want. Maybe I apply a little bit of heat to speed up the reaction, and boom, I have the product, and then you can purify the product out. But essentially, that's how you do chemistry in a lab. But in the cell, there's all these reactants floating around. And in the case of a condensation or hydrolysis reaction, one molecule that's involved in both of them, in the case of hydrolysis reaction, H2O is a reactant, remembering that a hydrolysis reaction is this. So one reactant is available in almost infinite amounts, and the opposite direction would be a dehydration reaction or condensation reaction where the byproduct is H2O. So this molecule, H2O, is available in almost infinite amounts. So in this environment, how do you control the reactions that are happening within the cell? Well, none of these reactions are going to happen very quickly without the help of enzymes. So a lot of these reactions without an enzyme involved would take 100 years to go to completion, 1,000 years to go to completion, etc. That's why in a chemistry lab, a lot of times you heat up the reaction pot to speed up the reaction. So in this case, you have enzymes. And if I have a reaction, A plus B gives me C, and there is an enzyme that is involved in this reaction, because there is going to be an enzyme involved in the reaction. Every reaction in the cell has an enzyme associated with it. Well, if I shut off this enzyme, this reaction is not going to happen. Now, a chemist will say, well, the reaction will happen if it's a favorable reaction, and we'll talk about what favorable reaction means later. If this reaction is favorable, meaning that it should make the product C, well, it's still happening. But because the enzyme is not involved, it's happening at such a slow rate that it is not significant to biology, meaning that it's happening at such a slow rate that it cannot support life. So it's like it's not even happening. But if the enzyme is active, then this reaction will happen very quickly. In a matter of fraction of a second, this enzyme will make this reaction go to completion, and you'll get your product. So when we say a cell controls the chemical reactions within it by controlling the activity of its enzymes, we mean that if I have a number of reactions, like A goes to B, B goes to C, C goes to D, and I have enzyme 1 for this reaction, enzyme 2 for this reaction, and enzyme 3 for this reaction. Well, if I don't want the first reaction, A goes to B, to happen, if the cell doesn't want that reaction to happen, it has plenty of B, it doesn't need to transfer any more A into B, all it has to do is shut off enzyme 1, and B will not be made. If it has plenty of C, so there's no more need to transform B into C, then all it has to do is shut off enzyme 2. And then you're not going to get any more B transformed to C. And the same true for C goes to D. So by controlling the enzymes, the activity of the enzymes, by simply having two states, whether the enzyme is active or inactive, and controlling that, the cell will control the chemical reactions happening within it. Will a chemical reaction happen in a cell if it is not catalyzed by an enzyme? And the answer to that is no. Remember, we're talking about biology. We're not talking about organic chemistry or just chemistry. We're talking about a chemical reaction must happen within the time frame that is important to maintain life. If it happens way too slowly to maintain life, it's like it doesn't even happen. So what are the methods by which a cell controls enzyme activity? Reversible inhibition. Remembering what reversible means. Reversible means that if a molecule binds to an enzyme and inactivates it or activates it further, that interaction can be reversed without the need of a chemical reaction. Meaning that interaction is a binding event that is mediated through weak interactions, hydrogen bonding, ionic bonding, etc. It doesn't require a chemical reaction for it to be reversed. So what does competitive inhibition look like? Let's say that we have a reaction, A goes to B, and we have an enzyme involved. So in this case, A is the substrate, the substrate is A, and the product is B. Here is our enzyme plus our substrate, and this gives us our enzyme substrate complex. And then we get our enzyme product complex. The product will look a little bit different, and then the enzyme releases the product. Now, what does a competitive inhibitor do? So a competitive inhibitor will bind to the active side of an enzyme. So here's our inhibitor, and this will give us an enzyme inhibitor complex. Now, as you can see, the inhibitor is wedged into the active site, so the substrate can no longer bind to the active site. So the inhibitor and the substrate are competing for the same site on the enzyme. And that's why it's called competitive inhibition. So this can be written out like this. So I'm just going to get rid of all the shapes and write it out. So we have, going from the top, enzyme plus substrate, giving me enzyme substrate, which gives me enzyme product, which then gives me enzyme plus product. Enzyme plus inhibitor gives me enzyme inhibitor. Now, if the concentration of substrate is increased, what will happen is the inhibitor can be kicked out by the substrate. And you can go directly from enzyme inhibitor to enzyme substrate and then get your product. So competitive inhibition, that's the hallmark of it, meaning that you can, by increasing the concentration of substrate, you can actually overcome the inhibition that the inhibitor is causing. Now, a few things to consider. Since this is reversible binding, instead of a straight arrow, I can put a double arrow here. And what does that tell me? That tells me if I lower the concentration of inhibitor, then I will also decrease the inhibition. So the whole thing is dependent on concentrations. And we'll get more into detail when we talk about binding in more detail later. But essentially, if we're ever talking about reversible binding, for example here, it's also a double arrow. Because it's reversible binding, the substrate can bind to the active site and then exit the active site without being turned into product. The same thing at this point right here. This is going to be reversible binding too. But the hallmark of what makes something a competitive inhibitor is that A, the inhibitor binds to the same site as the substrate, the active site, and B, that the inhibition can be overcome by increasing the concentration of substrate. And that's what this thing is right here. So if the question says, I have an inhibitor, and by increasing the concentration of the substrate, I can overcome the effects of the inhibitor, then you know right away that's reversible inhibition and it's competitive inhibition. So here we can simply write competitive inhibition to be enzyme plus substrate, gives me enzyme substrate, gives me enzyme product complex, gives me enzyme plus product, inhibitor, enzyme inhibitor, and I can overcome this inhibition by increasing the concentration of substrate. Non-competitive inhibition. So essentially for non-competitive inhibition, everything is almost like competitive inhibition. So we'll start from the beginning and draw it out. So my enzyme, here is my enzyme, plus the substrate, gives me the enzyme substrate complex. And this turns into enzyme product, and then the enzyme and the product dissociate from each other. Now in this case, the inhibitor binds to a different side of the enzyme rather than the active site. And by binding to this different site, it actually changes the active site so that it can no longer bind the substrate. So the inhibitor and the substrate are not competing for the same site on the enzyme. They are binding to different sites on the enzyme. So this can be written out like this. Enzyme plus substrate gives me enzyme substrate, gives me enzyme product, then the product is released, and plus inhibitor gives me enzyme inhibitor. But in this case, I don't have that arm that was going in this direction for competitive inhibition. Because if I increase the amount of substrate, there will be no effect. Why? Because the substrate and the inhibitor are not competing for the same site on the enzyme. So the only way to overcome this is that you decrease the concentration of inhibitor. So by decreasing the concentration of the inhibitor, you can overcome or lower the inhibition being caused. There's no other way. Increasing the amount of substrate will have no effect. So if we're going to write it here, you just write what we wrote before. Enzyme plus substrate gives me enzyme substrate, gives me enzyme product, and the product is released. And the inhibitor will create the enzyme inhibitor complex. And that's non-competitive inhibition, which is reversible. So, for example, a question that says that an inhibitor decreases the enzymatic activity of an enzyme or catalytic activity of an enzyme, and increasing the substrate concentration does not remove that inhibition or lower that inhibition. What type of inhibition is this? Then it's going to be non-competitive. Reversible activation. In this case, what you have is you actually have an activator that increases the activity of an enzyme. And there's plenty of examples of this in biology. So you have an activator that binds to the enzyme reversibly. You have an enzyme that binds substrate and gives you enzyme substrate, gives you enzyme product, that gives you enzyme plus product. Right? So the activator, if bound, so we'll call the activator A. So if A is bound to the enzyme, here's E with A bound, so we'll just call it EA. Plus the substrate, what you'll get is just a little bit more enzymatic activity. The enzyme will just simply work a little bit better. The enzyme's catalytic activity will increase, meaning that the rate by which it catalyzes the reaction increases. The enzyme is working a little bit better. And again, this is reversible, meaning that the activator is binding to the enzyme through weak interactions. It's not a covalent interaction. Irreversible inhibition. So an example of irreversible inhibition would be phosphorylating an enzyme. So an enzyme is on, but when it is phosphorylated, it is off, meaning it no longer works. So in this case, a covalent bond has been made. A phosphate has been covalently attached to the enzyme, putting it in the off position, and that's why it's irreversible, because you would need a second reaction. So the second reaction, I'm going to draw it here. E phosphate plus H2O gives me the enzyme back plus an inorganic phosphate. So now we go from off to back to being on. So these are two different reactions, and interestingly enough, both of these reactions, this process of turning an enzyme from the on to the off and then from off to on, have their own enzymes. So enzyme 1, enzyme 2. Irreversible activation. Irreversible activation is simply this. Meaning that you have an enzyme in the off position and you need a covalent interaction to either to turn it on from the off position. Now, in this case, dephosphorylation turns it on, but you could also have the enzyme is in the off position, but phosphorylating it will put it into the on position. And again, there'll be an enzyme catalyzing this reaction because this is a chemical reaction. You're making a covalent bond between the phosphate and the enzyme. Gene expression. Simply expressing the gene whose product is the enzyme. So you go from gene, getting the messenger RNA, and getting your enzyme, the protein. So the enzyme might not be in the cell. Something happens, the cell starts expressing the gene for that enzyme, and you get the protein, the enzyme being made, and then you have that catalytic activity in the cell at that point. All right, I think we'll stop here and continue later. Thank you.